Today we are going to talk about VLDL transport and metabolism. Okay, that is very low density lipoprotein transport and metabolism. Now, first of all, when I am talking about very low density lipoprotein transport and metabolism, you must first understand that VLDL transport and metabolism is actually conversion of VLDL to LDL. That is, the final product of this metabolism is conversion of VLDL to LDL. Okay, that's it. So our main goal is to convert this VLDL to LDL. Clear? Now, first important point you need to understand is this, that before you start this thing, okay, you must know what are these lipoproteins, what is VLDL, what is LDL, what is IDL, everything. If you don't know this, then go to the part 1 of this video series, you will understand. Those who are watching the video, I am telling them also, go to the part 1 of this video, then you will understand what are these things. Clear? Now listen. When we are talking about VLDL transport and metabolism, our main aim is to convert VLDL to LDL, right? Now listen. Now, if we are going to convert this VLDL to LDL, then we will have to first know that where does VLDL come from? Actually, VLDL comes from liver. Clear? It comes from liver. Clear? That is, VLDL is produced in the liver itself. VLDL or very low density lipoproteins are produced in the liver itself. Clear? Now, let's talk about its metabolism in a very much fast manner, because it's a very easy thing, an easy topic right now. Listen. When liver produces VLDL, okay, when liver produces VLDL, suppose this is your VLDL, okay, suppose this is your VLDL, okay, then you must know that VLDL is actually rich in triacylglycerol. It is rich in triacylglycerol, but also contains cholesterol plus cholesteryl ester. VLDL is rich in triacylglycerol, but it also contains cholesterol and cholesteryl ester. Clear? Okay. Now, most important thing you need to understand is this, that liver will help in the release of these VLDL, which is rich in TAG, and when it will actually, suppose this is your circulatory system, suppose this is your circulatory system, suppose this is your circulatory system, okay, clear? Then you must understand that when this VLDL enters circulation, which is rich in TAG, they contains a protein on their surface that is known by the name of ApoB100. Clear? It is known by the name of ApoB100. Unlike ApoB48, it is ApoB100. ApoB48 we have discussed in details in chylomicron metabolism and transport. Okay, this is ApoB100. Why it is B100? I am not going to talk about that, because we have discussed in details about the transcriptomics of this particular protein. Okay, now listen. When this tag-rich VLDL enters the circulatory system of our body, it lacks a very basic thing. What do you know? It lacks apolipoprotein C and apolipoprotein E. Now listen. Apolipoprotein C and apolipoprotein E, these two are contributed by HDL. They will transfer this apolipoprotein C and also they will transfer apolipoprotein E here. Clear? Now, when this tag-rich VLDL gets apolipoprotein C from HDL and apolipoprotein E from HDL, then it looks something like this, okay, somewhat like this. That is, clear? This is your HDL, which is giving C and E. Clear? That's it. Now, important point you need to understand is this, that when this VLDL will enter the circulation, then this VLDL is actually activated by, now listen, when this tag-rich VLDL enters the circulation, then the apolipoprotein C of VLDL will interact with this portion of this cell. What is this cell? What is the name of this cell? What is the name of this cell? These are endothelial cells, suppose. Now listen. So these cells are part of this blood vessel, okay? So now listen. So these endothelial cells have special proteins, we already know, that is lipoprotein lipase, right? Now listen. These lipoprotein lipases are activated, these lipoprotein lipases which are already present on the surface of endothelial cells are activated by apolipoprotein C. They act as a cofactor, okay? Now listen. When they are activated by apolipoprotein C, then they will try to act on this tag, that is triacylglycerol, and they will take up fatty acids and they will give out glycerol. Clear? They will give out glycerol. Now listen. Now after doing this, that means the concentration of tag is what? Decreasing, right? Now listen with all your attention. After this activation of lipoprotein lipase, what will happen is this particular thing is now listen. Concentration of tag is now lower, right? Now listen. When the concentration of tag is lower, then it is called LDL, right? Now listen, there is a very basic difference between LDL and IDL. Now listen. If you think that this is LDL or low density lipoprotein because concentration of tag has decreased, then you are wrong. This is called ideal, intermediate density lipoprotein. This is ideal. This is actually ideal. Why it is called ideal? Now listen with all your attention. Suppose this is your HDL, okay? Now HDL will again take back two things, apolipoprotein C and apolipoprotein E, which were its components. They will take back these things, E and C, okay? Now listen with all your attention. Now listen with all your attention possible. When the concentration of tag is lower, but apolipoprotein C and E are present, then it is ideal. But when concentration of tag is lower and apolipoprotein C and E are gone, they are called LDL. Now listen with all your attention. Important point you need to understand is this, that ideal have an unique thing. Okay, now listen. Very important point here you need to understand is this: ideal is very unique with respect to many things, but I will talk about this later. Let me talk about this. Suppose this is your blood vessel. Okay, so this is your blood vessel. Now listen, this is HDL. Now listen, this is tag. Okay, now listen, there is a protein, which has two sites. This is called CETP, cholesteryl ester transporter protein. Okay, now what it does is, actually this takes up cholesteryl ester from HDL and transfer it to IDL, and it takes up tag or triacylglycerol from IDL and transfer it to HDL. That means the whole and sole important function of CETP is this: it actually helps in it acts as a transporter for cholesteryl ester, and it transports cholesteryl ester from HDL to IDL, and it transport tags from IDL to HDL. Now listen with all your attention possible on this earth. So this is the function of this small protein, which is very important. Now what is so peculiar about IDL? IDL contains apolipoprotein E, right? Now listen, liver cells have receptor for apolipoprotein E, that is ApoE receptor. Liver cells have receptor for apolipoprotein E. Okay, now listen, apolipoprotein E, this apolipoprotein E are present in three isoforms: apolipoprotein E2, E3 and E4. Clear? E2, E3 and E4. Now important point you need to understand this again is the affinity of E2 is way lower. Affinity of E3 is the highest, and it is the most common, apolipoprotein E. Now listen, those individuals who have apolipoprotein E4, they have a natural tendency for Alzheimer's disease. Clear? Now this is associated with Alzheimer's disease. Why? We'll talk about the pathogenetic changes during Alzheimer's disease. We will have a separate video on that. Now listen, when this E4 is present, when any individual is homozygotic for this E4 protein, then they have increased chance for Alzheimer's disease. Okay, when any individual is homozygotic for this E2 protein, then E2 protein has, this E2 apolipoprotein has low affinity for ApoE receptors, low affinity for apolipoprotein E receptors. That means these protein, when any individual have two copies of these genes, apolipoprotein E2 genes, then their body cannot effectively clear IDL. That means they will have a high concentration of IDL in their body. But if you are homozygotic for this E3, if you have normal copies of E3, then you are safe. Okay, clear? Now listen, we'll have a separate video on these things, don't worry. Now listen, so what is the difference between IDL and LDL? IDL is containing B100, C and E, low concentration of tag. But when CETP acts, then it is no longer IDL, it is on the verge of becoming a LDL. Why? Because HDL has come and HDL will take C and E. Now listen, after becoming LDL, that is rich in what? Cholesterol ester plus tag and cholesterol. They have right now what? B100. Now this apolipoprotein B100 also has receptor in the liver cell. B100 receptor, and they are taken up by liver cells and they are metabolized. Clear? So this is in short the VLDL transport and metabolism. Clear? Okay.